Hi guys, Miss Francis here, and today we're going to be talking about another group of macromolecules known as proteins. Proteins are us. All of our proteins are responsible for just about everything that we're able to do. Our hemoglobin, which carries our oxygen through our body, is a polypeptide, a protein. Antibodies are proteins. They're going to recognize foreign pathogens and protect our bodies. Collagen in our skin is a protein. Collagen breaks down as we get older, and that's how we end up with wrinkles. Spider silk is a protein that is produced by spiders, and in fact it's so strong that research is being done on it to actually use it for protective gear for cops, bulletproof vests. The enzymes, such as Rubisco, are extremely important proteins. Enzymes are responsible for speeding up our chemical reactions. So what do these all have in common? They make us what we are. So here we go. Proteins are instrumental in about everything that an organism does, where the functions include growth and maintenance. Our hair, our fingernails are all proteins. Enzymes, hormones, and antibodies are proteins that play a regulatory role. So here's a nice little chart that summarizes all of the important functions of proteins. This chart is also available for you in your note packet. Proteins are extremely diverse. We have tens of thousands of different proteins, each with its own structure and function. And each human has its own set of proteins called their proteome, and each cell even has its own proteome. So here is a little video from Stephanie Castle where she discusses proteomes in a quick way. Each protein is made out of monomers known as amino acids. There are 20 different amino acids, where the polymers of these amino acids are called polypeptides. So a protein consists of one or more of these polypeptides folded into a specific conformation. So let's take a look at what makes up this polypeptide. So the polypeptide is a polymer of amino acids connected in a specific sequence, like we said, where that amino acid consists of 4 components attached to a central carbon. So here is our central carbon, and attached to that central carbon you should notice a familiar group. This is our carboxyl group. And remember, the carboxyl group has the ability to lose a hydrogen. On the other side, we've got a new, or our third functional group, called the amino group. And then you have this R group. The R group is what's going to vary among the amino acids, and that's going to dictate which amino acid we're looking at. Let's get into the amine group even deeper. So the amino group consists of a nitrogen atom attached to 2 hydrogen atoms. And the property that the amino group will impart on an organic compound is that it can act as a base because it can pick up a hydrogen ion. I mentioned there are 20 different amino acids. There are 8 or 9 essential amino acids that humans can't produce. We need to get them from our diet. So when we say we are what we eat, it's because we really are what we eat, right? Those proteins are going to be broken down into the amino acids and the essential chemicals that we're going to need for our chemical reactions. But The amino acids get their variety because of the different R groups, and these R groups can be hydrophobic, hydrophilic, or they can be charged. Stephanie Castle discusses the variety of amino acids in a nice short video. So let's get into the different groups of amino acids. Here we have the hydrophobic R groups. So the hydrophobic groups are going to be non-polar. They're not going to dissolve in aqueous solutions. Some important ones, methionine. Methionine is always going to be the first amino acid in any amino acid sequence. Phenylalanine. PKU is a disease where a person cannot break down phenylalanine, and the phenylalanine builds and builds and builds. However, this can be controlled by diet, so it's a fatal disease if it's not controlled, which is why every baby when they're born gets tested for PKU, because the simple cure is just to regulate that person's diet. They need to limit the amount of meat, dairy, beans, fish, anything that has a real high protein content in it. Another hydrophobic R group that you might be familiar with, or amino acid that you might be familiar with, is tryptophan. Tryptophan is found in turkey. It's that amino acid that makes us feel sleepy after we eat our turkey. The second group of amino acids that I'd like to talk about are polar amino acids. They are hydrophilic. They will dissolve in water. We'll come back to these in a second. The last group of amino acids includes those with functional groups that are charged or ionized, where some R groups are going to be bases and some are going to be acids. So the sulfhydryl group is a sulfur atom bonded to a hydrogen atom and to the backbone. Kind of resembles a hydroxyl group in shape. Instead of the OH, it's an SH, a sulfur, and a hydrogen atom. And the sulfhydryl group helps to stabilize the structure of the proteins. These groups will show up in what's known as the tertiary structure of our protein. So let me go back to an example of an amino acid that has this group. Cysteine has this sulfhydride group. All right, so let's keep going. Amino acids are going to be joined together through the process of dehydration synthesis. In order to join them, you need to form a bond between the neighboring amino acids. And the result is a covalent bond known as a peptide bond. So here we are removing a water and getting a peptide bond. So here's a peptide bond and here's another peptide bond. And the peptide bonds are formed between the amino acids, not within each amino acid. So see, I have 1, 2, 3 amino acids. Even though there's a nitrogen-carbon bond within the amino acid, that's not a peptide bond. The peptide bond is formed between 2 different amino acids. So make sure you are able to distinguish that. Stephanie Castle goes through the formation of polypeptides via dehydration synthesis. You guys are going to, at some point, be asked to draw 2 amino acids and then show how a polypeptide is formed via dehydration synthesis. So to start out drawing an amino acid, I would start with the central carbon. From there, we might want to draw our carboxyl group. Then we can draw our amino group. And lastly, we can draw our R chain and our hydrogen that is on the opposite side of our R chain. So here you see that we've got 2 amino acids. We've got 1 amino acid here and our second amino acid here. And we are going to remove our water, which is here. It's the hydroxyl group from 1 amino acid and a hydrogen off of the amino group from the neighboring amino acid, and that's going to give us our peptide bond. When we join 2 amino acids, we form a dipeptide. When we join 3 or more amino acids, we form a polypeptide. So here's a video which Stephanie Castle shows us how to draw an amino acid. And by repeating this process over and over and over again, by linking amino acid after amino acid after amino acid after amino acid, we create this long polypeptide chain. So let's take a look at some review questions. How are all amino acids the same? They all have a central carbon, they all have a carboxyl group, they all have an amino group, and they all have an H atom. The way they differ is by their R groups. And it's this R group that's going to determine the shape, which will then determine the function of the protein. So how can living organisms build such a large variety of polypeptides? It's because of that R group. Furthermore, you change the sequence of the amino acids in each chain as well. Proteins have 4 different structures, and that's what we're going to be talking about in our next lecture. So I'll see you then.